explains the functioning of our immune system, how our immune system works when it encounters a particular antigen. And before we dive into the details of the theory, there are a few key terms that we must know. So what is the B lymphocyte? So B lymphocytes are a type of white blood cells which are particularly responsible for producing the antibodies. There are two types of B lymphocytes, the plasma B lymphocytes and the memory B cells. So plasma B cells are the particular lymphocytes or the white blood cells which produce the antibodies on encountering a particular antigen. Then we have antigen. What is an antigen? Antigen is any foreign substance or a toxin which has the ability to bind to the antibodies produced by the B lymphocytes, and it may or may not have the property of immunogenicity, that is to elicit an immune response. Then what is an antibody? So as I said, antibodies are the glycoproteins. These are glycoproteins in nature. These are the glycoproteins produced by the plasma B cells, and these are specific in nature. That means that a particular antibody will only bind to a particular antigen when the antigen will enter the body, and these are specifically produced by the plasma B cells. So the clonal selection theory, as I said, is a theory which explains the functioning of our immune system, that is the lymphocytes in response to specific antigen invading the body. So when any foreign antigen, suppose a virus such as the COVID virus or snake poison, scorpion poison, anything enters our body, as we all know, there are mechanisms in our body that will fight those antigens to protect our body. So the clonal selection theory explains how this mechanism works. So it is not, particularly, it doesn't start functioning only when we encounter the antigen. The cells are present in our body from before, but the antigen only proliferates those cells and causes an large response to fight it in the minimal time possible, which is not possible every time, but they tried. And it was proposed by Frank McFarlane Burnett in the year 1957, and he attempted to explain the great diversity of immunoglobulins or the antibodies which are present in our body. So he wanted to explain why we have so many antibodies already in our body. So which is why he encountered this theory and proposed it in the year 1957. So starting, as I said, the cells are present in our body from before. So different beta lymphocytes are not present, but they are produced from the lymphoid stem cell. So in our bone marrow, there are lymphoid stem cells which will develop into beta cells, and these beta cells will mature in the bone marrow itself in mammals and humans, but in birds they mature in buds of fabricus, which is why they are named beta cells, not because of the bone marrow. They are named B cells because they mature in buds of fabricus in birds. So the beta lymphocytes will mature in the bone marrow, and then they will get transferred to different lymphoid organs such as the spleen, then we have lymph nodes, then we have appendix and Peyer's patches in small intestine, and many others. So when the antigen will enter our body, it will get mixed with our body fluids, and there it will encounter those antibodies which will fight against it. So beta lymphocytes are differentiated in the bone marrow, and they are programmed genetically to produce a particular type of antibody. So one beta lymphocyte will only produce a particular kind of antibody which will be specific to one kind of antigen. So they will produce those antibodies, and the antibodies are, as I said, glycoproteins in nature. They are also known as immunoglobulins. They have this Y structure with heavy chain and light chain. So if you divide it can be divided into two particular fragments. This is the hinge region, which is rich in proline, which lacks in secondary structure, so it is really cleavable. It is susceptible to different enzymes such as papain and pepsin. So if we cleave it from here, this is known as the FAB fragment. This is where the antigen will bind. So these fragments are really specific, and this is known as the FC fragment, which is the crystallizable fragment. Here are the different crystallized complement proteins will bind in order to eliminate the antigen. So this may be similar in many antibodies, but these fragments are really specific. So every beta lymphocyte will have a particular FAB fragment, and these FAB fragments are where they are present. They are present on the surface of the beta lymphocyte. This diagram represents everything. So this is the lymphoid stem cell. This will mature into beta lymphocyte, and through gene translocation, that is immunoglobulin gene arrangements, which is a completely different process. Let me know if you want to know. So they will produce different kinds of antibodies. I don't know if it is visible, but the receptors are different in all the three beta cells. So every beta cell will have a specific receptor which will be on their surface, and it is also known as the beta cell receptor or the antibody. And when the antigen will enter the immune system, this is an example of an antigen. Suppose a virus, it will have different epitopes. So these epitopes will bind only to a particular kind of antibody. This is an antibody, this is an antibody, this is another antibody. Say this is only specific to this one, so it will go and bind to this one. Now this will activate the beta lymphocyte, the binding. Okay, so the beta lymphocyte will get activated on encountering a particular antigen. So the activated beta lymphocyte will now proliferate. The affected T lymphocyte helps in proliferating the beta cell by producing cytokines. This is the mechanism of humoral immunity. So it will proliferate into a large population of the same clones of the beta lymphocyte, which will bear the exact same copy of the antibodies. So these are the same cells, but in very huge amount to encounter the antigen, to fight with those antigens. So what they will do, these antibodies will bind to the epitope and they will either tag the antigen to carry out an immune response to protect the body and eliminate or neutralize the antigen. By this, what I mean is, say this is an antigen, the beta cell will either tag it. The beta cells do not kill those antigens. They are not capable of that. They will either tag the antigen and they will present it to certain different phagocytic cells such as macrophages and neutrophils. They will kill the antigen. Or by neutralizing, I mean, this is suppose an antigen. They will bind to the surface and form a layer. So the viruses need to communicate with other cells in order to cause that effect. So they won't be able to communicate with the other cells. Or say, suppose this is a toxin, some snake poison or scorpion poison. So the snake poison or any poison has to also interact with certain molecules in order to elicit its response, but they won't be able since there is a layer of the antibody. So they will either eliminate by using with the help of macrophages and neutrophils, or they will neutralize the antigen. So if you just pay attention to this is the entire process of the clonalization given in a single image. So those are the beta lymphocytes. Every beta lymphocyte has a separate and a distinct beta cell receptor. These are present in our lymph, that is the body fluid. So when an antigen enters, it will bind to only one particular receptor, say this one. It will bind to this, and now it will activate the cell. This is known as clonal selection, or this is known as clonal selection. This is the clonal proliferation. What means? This means that the activated cell will now proliferate into a number of cells, and the number of cells can be divided into two distinct types, which are the memory cells. Now, mind, every cell will have the same receptor, since these are the identical copy of the parent cell, and there will be two different types. One will be the plasma B cell, which are responsible for producing the immunoglobulins for the antibodies, and there will be memory cells, which have much longer lifespan. Then the plasma cells will literally work till death. They will work and then they will die. They will produce the antibodies, they will do their duty and then they will die. But the memory cells will stay since they have a very long lifespan. So the memory cells will be stored and the beta cells will form the plasma B lymphocytes and then will produce the antibodies which will either eliminate or neutralize the antibodies. So there are these few postulates about the clonal selection theory. So as I said, there are a large variety of lymphocytes which are present in our body which can bound to a tremendous range of antigenic epitopes. This is why a large number of lymphocytes are present, so that we may encounter any disease. So there has to be variety. So this is why tremendous range is present, and each lymphocyte but only bears only one single receptor, as I said, which is unique in specificity to a particular antigen. So which is why there may be some disease which we do not have the antibodies for, because it has to be specific. If we have, say, 1 lakh antibodies, but we don't have that specific type, so we'll get the coronavirus. So because these are specific in nature, and then the receptor occupation will bind to the lymphocyte is required for, and it will activate the lymphocyte, which is known as clonal selection, and the differentiated effector cell will produce identical copies of the same parental cell, which will have the exact same copy of the beta cell receptor or the antibody. And these will be and those lymphocytes which have the antibody specific to our own cells. So it may cause an immune response which is self-destructive, so they will be deleted at an early stage, so that we don't have any harmful antibodies in our body. So now I would like to conclude my presentation with a few concluding points. So an antigen therefore selectively stimulates only those cells which have the complementary receptor